This is my Tuesday seminar. So a little bit about myself. I've been here for 3 years, and my position, I'll talk about that in a little bit more detail in a second, but basically it's 80% teaching, 20% research in the SOTL field, or scholarship of teaching and learning. So when Mark asked, excuse me for the clock, when Mark asked for a title earlier this semester, I gave him this relatively generic sort of a title. But in truth, what I thought I'd like to do is kind of bookend a couple of different things. At the end of last semester, Felicitas from Grandview gave a really nice talk about teaching at a liberal arts school for kind of like an alternate sort of career path. And so I wanted to talk a little bit about the relationship between that. For me, teaching here at a land-grant university is kind of a different career path, as well as to bookend that with a brown bag a couple of weeks ago. Jeff Iles, the chairman of the horticulture department, gave a really nice talk about what it's like to be the chairman for a day. And so I kind of wanted to also give a little bit of information about what is it really like, what do I actually do on a day-to-day basis. So I'm going to talk about teaching at a land-grant university, the alternate career path, and a day in my life. Thanks. All right. So kind of the roadmap for what I'm going to talk about. So what is my background itself? What does my actual position entail? What's kind of like a normal day like? What sort of classes do I teach? How do you actually get started in the SOTL research field itself? Can you actually do SOTL research in a wet lab sort of a setting? And then finish up with what I perceive are some of the bigger benefits of my position. Maybe not so much the concerns, but what are some areas for extra attention to pay attention to? And then where things stand out in my third year review, and then maybe a little bit about some future plans. So please ask any questions as we go through throughout the entire talk itself. All right, so what's my background itself? So I did a bachelor's degree in zoology, which was basically just biology at Miami University in Ohio. In 2001 is when I graduated. So I didn't really get started in research until relatively late in my career as a undergrad, and so I decided to stay there for a master's degree in Drosophila genetics, and that also gave me the time to figure out what sort of things I wanted to do next for a PhD sort of a position career. And in that pathway, at the times I was applying to schools, I mostly applied to schools in the Midwest, and my advisor at the time suggested just apply somewhere else, one place outside of the Midwest, which is a good suggestion. In the end, I asked him if he, do you have anything in mind? And he had some connections with the University of Utah. So I thought, all right, it's a free application, I'll apply there. In the end, it turned out that I really, really enjoyed it there, and that's why I ended up going for my PhD. My PhD from Utah is actually in cellular and developmental biology in relationship to how brown owl zygotes establish a polar axis and make their first asymmetric cell division. Somewhat ironically to that is that the organism I worked on is a stromatophile related to the oomycetes that we talk about as plant pathogens. Completely ironic that it worked out that way. And then from there, right towards the end of my PhD, I actually had a fellowship work in a marine biology station in Roscoff, France, which is in Brittany. And during that time, which was really, really fun to be there, it seemed to me that probably the field of research in this type of brown owl zygote not going to have a really long-term future. And so I was thinking about other sorts of career options which might have a little bit more funding, a little bit more areas of interest. And so what turned out to be really beneficial to me is when I came back to the fall semester, one of the people giving just like a regular talk like this, for us it was Friday talks, was a guy named Tom Bernhardt from Harvard Medical School. And he gave his talk on basically how bacterial cells mediate the cell separation process. How does one bacterial cell remodel the cell wall and the membrane to become two different bacterial cells? And so I thought that was fairly interesting, since a lot of the stuff I was working on was how one cell makes an asymmetric cell division choice leading to two different cells. And so I had lunch with him. We kind of kicked around some different ideas about what his work was on and some of my interests. So I asked him if he was actually taking any postdocs. And he said, well, it depends on whether or not this first NIH grant that I'm writing gets funded. So thankfully it did get funded, and ultimately he decided to hire me at the end of 2009, beginning of 2010. And so there I worked on cell separation in bacteria, and then ultimately, as you all know, I started here in January of 2016. So kind of an interesting sort of pathway leading up to a position of 80% teaching, 20% research. All right. So one thing's actually truly, what is my position responsibility statement, or the actual contract I have for the first 3 and then the second 3 for the 6 years towards tenure. So it's a tenure track position, retention review, which is underway right now after 3 years, and then the tenure process after 6 years. Also at the time, I really didn't know what a 9-month appointment versus a 12-month appointment even really meant. But in the end, in some respects, it's kind of nice from the standpoint that I don't have any teaching responsibilities over the summer. Clearly that doesn't mean that I don't have to work. There's still all sorts of additional things in the lab with other students, manuscript writing, going to conferences, stuff like that. But while you also don't have any teaching, you also don't get paid over the summer, so maybe that's a slight minus on that one. As far as teaching and advising, so the real core of my position. So I should teach between 5 and 7 credits for every semester, and then for advising, up to 40 total students for advising. Advising is kind of an interesting one because right now I have 19 advisees. It start off with 5 or 6, and then slowly Joan, who's in charge of the micro programs, increased that number up to 19. But advising itself is kind of like an all or none sort of a proposition, because right now, this week and next week, is like the season of advising. So all 19 advisees need, under whatever their schedule fits out, at least an hour to talk about their schedule, their classes. And so 19 is fairly manageable. Working up to 40 will still, it'll be difficult but manageable. But I can only imagine what having, what was the height number?
My highest number, 105. When I started this position, 83.
Yeah, so 83 advisees is extreme. So thankfully, I may have to go up to 40. And then for research, 20% of my work, and it has to have some flavor of scholarship of teaching and learning. And that's defined as peer-reviewed publications, which is fairly obvious to people, teaching modules, case studies, videos, other sorts of projects and products, and then service. Service actually works out for me relatively nice, that 3 of these really tie together pretty clearly. The CALS Curriculum Committee, then just being a member of the Departmental Curriculum Committee, and then the catalog editor, all 3 of those kind of tie together. And so we can deal with at the college level what the curriculum decisions they're making, and I'm the representative for Plant Path and Micro, and then can share that information back with our curriculum committee here, and then changes that we make to the catalog, then I'm in charge for submitting all of those. And then additional things like the awards committee, that's actually, it's kind of busy towards, most awards are due in kind of like the middle of November, but it's kind of fun to see what the department, who people are applying for different awards, who's being set up for different awards and stuff like that. And then also the library rep and co-advisor for the micro grad student organization, which don't really take very much time at all. All right, so that's kind of what I'm expected to do, kind of my contract of my sort of work. So over here's kind of like I consider to be relatively normal sort of a work week from a couple weeks ago. Obviously this week is mostly filled up with all sorts of advising sort of mumbo jumbo. But from a normal work week, here in green you can see all of the time in which I'm actually in either a lecture hall or in a laboratory with different students. So it's about 14 hours a week in which I'm directly hands-on with the students themselves. And then I fit all the other stuff in around that sort of a time frame. So the other sorts of things that are common, of course, like today's seminars, as well as our excellent brown bag on Fridays at lunchtime. And also in this week, we had the CALS Curriculum Committee meeting this day, a micro review session for their exam coming up. And also, once in a while, let's say 4 or 5 times a year, the micro program has high school students come in to get a feel for different sort of microbial activities, sorts of different techniques on Saturdays. And so that's maybe not when my alarm goes off. I'm not super interested in that, but the students really, really enjoy it. And that's a lot of fun to see what they do. And then the rest of the stuff are filled in with all sorts of sometimes reoccurring student meetings from different courses, or other students that are working in the lab that need other sorts of stuff to deal with. And then all the additional sorts of parts of the job are then squished in either beforehand, afterhand, or sometimes in between. I'll definitely say for sure is that usually by the end of the day is Thursday, by that point. All right, so what sort of stuff do I teach? So one course that I teach and co-teach it with Nancy Bory is Microbiology 115, which is phage discovery. and Microbiology 116, which is phage genome annotation. So the setup and kind of the background for that course is organized through the Howard Hughes Medical Institute. So Howard Hughes was a relatively famous aviator, inventor, and owned Hughes Aircraft for a long time. And so he ended up setting up HHMI as a tax shelter to take the profits from Hughes Aviation and kind of hide them from taxes. So for decades he would fight with the federal government about, is it really inappropriate for a medical institute or not? Is it really a nonprofit that we don't have to tax? But in the mid-70s, he actually somewhat unexpectedly died. And when he died, he had no will, no relatives, no next of kin, and he basically left everything up in the air. So the courts fought it out for a couple of years, and they decided to put everything into HHMI. They then immediately decided to sell off Hughes Aircraft, and so then they were left, HHMI, with about $5 billion. And so what they decided to do with that is to then really focus on true medical research. And so right now the endowment for HHMI is about $22 or $23 billion, and they give out about $850 million every year to scientific research. Beyond just principal, primary research, and applied research, they also do stuff like programs related to education of science. And one of those, called the C program, or Science Education Alliance, is set up to try to help foster additional understanding of science from a really young age of undergraduates. So about 10 years ago, a guy named Graham Hatfield from Pittsburgh decided to write a grant for HHMI to set up this sort of a course, a fall course of phage discovery, and then a spring course of phage genome annotation. So he was a phage researcher, scientist, and he wanted to set this up so that you could actually have students themselves go with basically no experience in the research field before that. Go isolate soil samples, water samples, find phage in those, characterize them, and then have those genomes sequenced, annotated, and then actively in part of the actual, ultimately for the greater breadth of knowledge. And all these things are submitted to the National Repository for the phage in Pittsburgh, as well as GeneBank. So it all goes in there. The students are all authors on the submission. So why Graham was really interested in this is that not a ton is really known about phage ecology or some of the evolution of different phage. So I'm not sure if everyone here knows what a phage is. A phage is just a virus that only infects bacteria. And so a fairly interesting factoid about that is there's about 100 million times more phage on Earth than there are stars in our universe. So relatively large number of phage. And it turns over in the oceans, principally in the oceans, about half of all bacteria are turned over every 24 hours by phage infections, which lyse those cells. But not a lot is truly known about them. And so that's why he set up this course. And then shortly, after I started here, I talked with Nancy about it. I knew this course had been about 9 years so far in the making. And so, not that Nancy didn't already have enough to do, but I twisted her on to see if she was willing to be a co-instructor at this course, and she was, and so we started off not this school year, but the school year before in this program itself. So what do the students really do? In the fall semester, they go, the first day of class, they get a little baggie with some 50 mil conicals in it. They go get some soil, water, mulch from campus. Facilities is approved of all of that. And then they come back, we add phage buffer to that, and just filter sterilize them with 0.22 micron filters. And what's left is hopefully the viruses that are pushed through there. And then all they do is take that liquid, mix it with some bacteria, and a little bit of what's called top agar, or very soft agar itself, pour that onto a petri dish, and then look the next day and see if they actually do, did they find any phage. And so in this case, you can see it's kind of the reverse of a normal bacterial plate, in which here you can see bacteria everywhere, and all the little holes are where it started off with one individual phage infected one bacteria, then that lysed that bacteria, and then the 100 or so more phage infected the bacteria around it until you get these relatively round plaques.
How did you decide what bacterium you were going to use?
Yeah, that's a great question. So they started off with, their interest principally was Mycobacterium smegmatis. They wanted to stay in the actinomycetes, and they thought that smegmatis would be a good one because it's a sister species to tuberculosis. And so now, after about 9 years, they've decided we're starting to find some of the same phage over and over again. And so they wanted to branch out into other ones. So we use Microbacterium foliorum, which is just a soil-dwelling microbe. An interesting kind of sideline to that, one of Graham's hope is that maybe you could actually use these phage which infect smegmatis. Maybe you can find some of these phage that also kill tuberculosis. And what he hasn't published yet, but is hopefully will be coming out soon, is that several people actually had very recalcitrant infections in the UK. So they got jumped through all the different hoops and were able to give them actually these phage cocktails that they tested at Pittsburgh on tuberculosis that they found were effective against those. And just kind of like trial and error work of injecting how many phages you inject into someone's arm at a time, and it saved this one person's life. So that was kind of the premise for what they want to do, and now they're more interested in kind of the evolutionary tree of all these different phage. You can do it with anything, though. I'll talk a little bit later about how I'm doing with other bacteria. So the hope is wherever you can find bacteria, you should be able to find the viruses that kill them. Does that answer it for you?
Yeah, I was just thinking that I don't know much about phages, but are they relatively host specific or?
So for the most part, they're pretty host specific, even to the point of being like species specific, even to like subspecies of different strains of bacteria. But some are a little bit more promiscuous, and those are the ones they were hoping for. Turned out to be the ones that can kill SMEG as well as TB, as well as some of the other paratuberculosis as well. But for the most part, they're pretty much spot on to that particular bacteria, which might make them, as they are hoping for, a nice alternative to antibiotics. So a cool part about this course is that the students think it's kind of fun to go out and look for this soil stuff, but they're truly getting to learn a little bit about the real research sort of aspect of this. And as you might imagine, not everybody actually finds phage their first time, so they failed, they got to go back, get more soil samples. Maybe they find the phage, but then they go to try to purify them and they lose them, so they got to go backwards. And so for a lot of the students, they never anticipated that sort of a method really is how things really go in research. They're much more used to kind of a cookbook sort of recipe. And so some people really enjoy the real research, and some of the other students, sometimes the pre-med sort of students, find that completely unacceptable. They've done it once, it should work, and that's it. After that, their hands are off. So anyway, they work through all that, and then, so we isolate the phage. Thankfully, with a huge help from Tracy over in the Egan facility. She comes over, gives them a little tutorial, they set up their grids. She zaps them on the electron microscope, and then they come, get to see all their little phage. They get to name their own phage. And then they try to get a really high volume of those phage in a lysate, because then we're going to isolate the DNA, make sure that DNA is relatively pure. So Mahir down here is running his little agarose gel after we did some restriction digests. And then the class votes on 2 of the phage to then be sequenced. And Howard Hughes pays for the sequencing over the winter break. And then when we come back from the winter break, they actually have the FASTA file itself of the 2 different phage. And then the students basically take those FASTA files, put them into DNA Master, and use all sorts of different programs to try to figure out where are all the starts and stops. Are these truly genes? Are these not genes? Does HH-Pred or BLAST ever suggest this as a gene? And for them it's kind of interesting, and for me it's kind of interesting too, is because roughly only a third of the genes, or prospective genes in phage, have any known homolog anywhere. 70% or so are completely genes of unknown function. So it's a whole unknown world of what all those different genes really do. The structural genes are fairly clear, but any of the additional genes, not very obvious. So they go through all of that, and then the spring semester finishes up with them presenting their work at the Freshman Research Symposia. And then, again, they vote for 2 people to then go with an instructor to the national conference in the summertime. And so Howard Hughes pays for everyone's flights and lodging and everything like that. And this last year, Gretchen and Ina presented their poster, and you can see here at Janella Farms, the HHMI research facility outside of Washington, D.C., about 150 or so people there. The entire C-phage group is 120 or so schools spread out throughout the entire world, and thousands of different students that come through it. So a pretty cool course itself, to give people a real flavor of what some real sort of research is like at the undergraduate level. All right, so another course that they teach, Introduction to Microbiology. Every semester, it's our largest course that microbiology offers, up to 250 students a semester. And it's kind of a mix. It's a service course, so it's all for non-majors. And so it's mostly animal science, people involved in kinetics, kinesiology, those sort of environmental microbiology, environmental engineering, those sorts of associated fields. So one of the benefits to that course is that I really get the first chance, and sometimes the only chance, to explain how microbes are involved in every aspect of our daily lives, throughout every moment of it. But as you might imagine, being a service course, the vast majority of people aren't really super interested in that. And a big chunk of them are animal science trying to go into vet school and not really that interested, which is kind of curious to me since they're going to spend a lot of time as a vet dealing with microbes. But nevertheless. So this is probably representative about how big the classroom is. I would hope this is not my actual classroom, but I hope that this is what it looks like as I look out into the audience. But one of the drawbacks is many times it instead appears like this. So I'm standing there looking around and people are playing around on their phones and other things like that. So some of the technology stuff can be a benefit as well as a drawback. One of the nice parts of the technology that we use, like the new Top Hat response system, is that at the end of every class I'll ask 5 questions to all the students that review what did we talk about in the last lecture. So a couple of softballs, were you even here at all? And then several which are actually exam questions. And then one of the nice parts is I can finish off the class itself by asking an open-ended question, which is anonymous to them. And they can type in, and all I ask is, what do you want to review next time? So they type in whatever it is that didn't make a lot of sense to them or what they were uncomfortable raising their hand in class about. And then I can look through those after class and then start the next class off to refresh a little bit of that stuff. Because it's not always obvious to me the parts where they're missing that part of the lecture or things that don't make sense. And so it seems to be a kind of a nice way to do that. One of the drawbacks to that sort of a method, though, is that I tell the students, if there's nothing you want to review, you can just say whatever you want to say, not applicable, just put in anything and tell me a knock-knock joke or whatever. So people put in, random sort of stuff sometimes, which is fine. So the first semester I did this, it was about this time in the semester, and the students, I was starting to get these weird sort of responses of students. They kept asking these questions about what's the name of your pet lobster? What do you feed your pet lobster? Do you keep your pet lobster in your office? And I had no idea what these people were talking about. But it persisted and persisted. And then towards the end of the semester, one of the students, he comes into my office asking for help for the final. He says, oh, I was hoping I could see your pet lobster in here. I'm like, what are you talking about? What is your pet lobster? And so he's like, oh, this pet lobster thing, if you go on to rate your professor, there's a review in there on you, and it says, oh, if you ask him about his pet lobster, he'll give you extra credit. So I'm like, oh, thanks for whoever put that in. So now, every semester, this time of the year, this halfway through the semester, I have to explain to them, I don't have a pet lobster, I didn't eat my pet lobster, I'm not sure I didn't eat my pet lobster. But the students seem to be relatively amused, and they never let up on the pet lobsters. All right. And the other principal class that I teach is the Principles of Plant Pathology, 408 5-week. That's also every semester. And that's the largest plant path course that we offer here. It's usually up to 60 or so students. So that one we have really, really nice new facilities for the teaching lab as shown by this picture here with all new stuff, all new microscopes. Every one of the microscopes has their own wireless digital camera. So the students look at something onto the dissection or compound scope, they can take a picture, immediately download it to their phone or their computer, have that. And so one kind of a goal for that is to start to have them make sort of portfolios and things like that with that information instead of just little sorts of drawings. In some respects, I felt a little bit guilty moving directly into this space after Ed had, over in Betsy, such a long time in 231E, which is not quite as nice. For me, I think in the end, my biggest challenge of all for all the courses is up in the greenhouse to actually make all the different plants sick for the actual courses. You think I have the pathogen, I have the plant, it's temperature controlled, I should just be able to make them sick, but no, try to give them GOS as well. Sometimes it works, sometimes it doesn't. All sorts of different soybean diseases. The black rot and cabbage, that one always seems to work pretty well. And then tobacco mosaic virus. Tobacco mosaic virus is kind of an interesting one from the standpoint that people always wonder or ask, why, once you try to give these plants tobacco mosaic virus, why do you keep them down here in the lab room under a light bank? Why don't you just put them back in the greenhouse? Because I tell them that certainly in the greenhouse, because it can affect so many different species of plants, they don't want tobacco mosaic virus up in the greenhouse. And it's actually one reason by a lot of greenhouses, they kind of hope people are not going to be tobacco users because there's a fair amount of active viral particles in just regular tobacco. So I thought it'd be kind of a fun idea as we start to do our serological tests is just go and get some cigarette tobacco and show them. You can find plenty of tobacco mosaic virus particles in the tobacco, and they can just take the tobacco and rub it on plants. It'll give them tobacco mosaic virus. So for that particular one, I went to one of the local smoke shops here in Ames, and so I pull in, and there's a lady sitting out front smoking a cigarette, and so I walk up, and she puts the cigarette out and walks in with me, and we walk up to the counter, and I'm like, hey, do you just sell like regular, like loose cigarettes, like 1 or 2 cigarettes? And she's like, no, we don't do that. And I was like, well, here's the deal. I teach this plant pathology course. I know it expressed on these people that it's really easy to spread tobacco mosaic virus. The lady looks at me and she goes, oh, one more thing I need to worry about. Now I'm So I thought, well, I probably should tell her that it's not a human pathogen. But I was like, well, if it helps her quit smoking, But she ended up giving me a pack of cigarettes that they damaged somehow, so it works really efficient. All right, so how did I get started in soil research? So my postdoc was all involved in cell separation means in E. coli, and then trying to figure out what was falling apart for the cell division machinery. So I did a lot of making fluorescent fusions, live cell imaging, to try to see the mechanistic movement of the divisome as these cells try to grow and divide. How did I really get started then in soil research, since that wasn't my classical background? Before that. So I mentioned in the first slide, discipline-based educational research, and then scholarship of teaching and learning. So my understanding a little bit different of what those two are related to. The soil field is really trying to do things to improve your own sort of teaching through innovations, and then disseminate that to the greater community so they can maybe try those things. Whereas the discipline-based research is more to test theories that are out there to see if you can find generalizable findings for teaching, learning, different ways of thinking in those disciplines. So kind of all this work falls somewhere in that sort of a continuum. All right, so how did I really get started in sort of research? So the nice part was that pretty soon after I got here, Thomas asked if I wanted to work on one of his broader impact projects for a grant. And this was a project that he'd had running for a couple years with, it was called the Trouble of Grain Leaf Farms, a case study, lessons in biological, agriculture, and environmental sciences. And so it was Thomas with Craig, tons of people here in Plant Path and Iowa Public TV. So I already had these really nice videos, and it's a really nice framework for this case study, and was looking for some, if I was willing to work with them to kind of bring it to the last step. Make the tools necessary to assess pre and post of the case study, and then bring it to the publication sort of a format. And so for that, one of the things we were trying to keep in mind, because this was geared towards high school students, is how is this going to align with the next generation of science standards, which is basically a consortium of states and people who put together what they think what sort of things students should be doing, what they should know, how they should think, like how they should apply biology to mathematics and other sorts of components. And then also, can we actually get this kind of a clearinghouse called EQUIP to evaluate our case study and say that yes, this covers all the things that we wanted to cover for a next generation of science standards. So when it's published, it'll hopefully have much broader dissemination. All right, so one interesting thing about working, obviously if we're going to work with and try to do research on minors, I have to do an IRB approval for that. And for minors, it is exceptionally more complicated. So we have to do all the things for the IRB package itself. It's 30-some pages. I have to show them the pre, the post, all the certificates, the letter from the principal, all the things, the documentation to show that we're protecting the documentation. So all these things have to be filled out before you can do any of the research. It just takes time as it slows that sort of a product down. But thankfully, as of right now, Craig is almost done with our beta test. And so the next thing we're going to do is make any changes we think might be necessary, and then write it up for publication and submit it to EQUIP, and hopefully they'll give us their highest rating to show that it does achieve all these things that we want to from a case study. And soil research was a project that was actually started with Mark. And so Mark was interested in trying to understand, as he teaches the RCR sort of training courses, he was wondering, are the RCR courses, responsible conduct of research, are those more effective when you do it face-to-face, or is the city training, the online training, just as effective? So we started off with, and he wondered if Nancy and myself wanted to help him with that project. So we started off, and we made it through the Iowa State round, because you can only send one of these grants forward. And then shortly thereafter, Mark had to leave the project, so I've shaded him here. And so Nancy and myself, we thought, well, kind of have two routes. We can either basically fold up and not go on, since neither of us teach RCR. We've taken multiple RCR courses, but have never taught it. Or we can try to find a team which might help us get funding. So we teamed up with Clark Wolf, who's the director of bioethics here. At that time, he basically said, I'm willing to do it with you, but I have no time to write the grant the next month or so before it's due. And so we thought, well, let's still give it a shot, because if we do get it accepted, he'll then take over the PI responsibilities, which would make more sense since he's the director of bioethics. We also teamed up with Patrick Armstrong, who's an expert in psychometrics, which is trying to understand how, when you do surveys for people, how do you not do things that accidentally trick them into different flaws in logic and stuff like that. So our aim is ultimately to create a validated and reliable assessment tool, which we've named after Iowa State, or the ISART, to see what are the learning gains of taking different types of RCR sort of training. So where are we in that process? So this is our little Gantt chart of where we are. We'll start off with, of course, the grant is funded. Start off with establishing what are the learning objectives, and then we send what we think are most important concepts out to professionals who teach RCR and see if they have the same idea as us. And then we wrote little vignettes to see, okay, we try to think about big data management. You know, this person goes into their lab, they decide, oh, this data would be a lot better if I just throw these data points out. Can I just do that? And then then students will come back with open-ended answers that we'll then code and try to say, what were their misconceptions? The ultimate goal is a multiple choice survey which people can use with fairly high reliability and validity to show that, yes, these areas you're teaching, you have very good success. These other areas, maybe you don't even teach this one aspect of RCR. So you wouldn't expect any change from pre and post of the course itself. And so we are right now, right at this phase in the spring of 2019. Just getting ready to hopefully start writing some of our multiple choice questions after we clean up the vignettes. All right. And also, what about doing sort of research in an actual wet lab? So one huge benefit of being here was the standpoint that when the department moved from Bessie and other people moved from Science 1 over here into ATRB, that opened up some space for me to actually get some actual wet lab space, which is, my assumption is not particularly common when your actual position is 80% teaching and 20% research and so on. So it was a huge benefit that actually got some wet lab space. And so here you can see Larry's old lab here with all sorts of water baths, centrifuges, spectrophotometers, microscopes, glassware, chemicals, flow hoods, all of that sort of stuff. And so the nice part is, too, as you might imagine, starting off, if you're going to do research, and sort of research, you're probably not going to start off with a massive amount of startup funds. And so it's one way is that all this equipment, everything you can see, was completely for free. Most of it was from Addis and donated it to me. But also people left Bessie, a lot of equipment people just wanted to triage, it's still functional. So all those things you can piece together to actually put a relatively useful lab in place. All right, so what is some of the wet lab research we want to do? So we're going to look at one project for the C-phages course. We want to optimize the growth conditions for the bacteria that we use for that class. And then a little bit of additional work is I want to discover some other soft rot causing bacteria, soft rots or bacteria which cause soft rots in certain plants. And so, as Gary asked earlier, so these phage, the hope is, if I can actually find a whole bunch of these soft rot causing bacteria, then maybe I can find phage that can kill them, and we can use those for remedial sort of a measure for a lot of the crops that have lots of loss after you've harvested them, like carrots, potatoes, onions, things like that. And as of right now, in the lab itself, I have 5 undergrads that basically start off with very little lab experience and then a shared graduate student with Nancy. All right, so what are we doing for the C-phage course and the C-phage community? So some things that aren't known is how long does this bacteria itself remain viable? So if you grow it up and then put it in the fridge, does it live for about a week or 2 weeks, or when does it start to die? It's just not known. It would help the community if people know how long they can keep these bacteria for still maximal infection. Also, if you're going to produce these phage, you have to soak these plates with phage buffer. How long do you need to do that for to get maximum release of the phage particles? That's not known, and we're using undergrads to test that. As well as sometimes, which is kind of curious to me, is some of the plaques themselves from the phage make these really abnormal shapes, which is not very common. We want to understand why is that the case. People have assumed for a long time one phage should always make one very discrete shape and size plaque. Cloudy, round, really big, really small, whatever, but they should be uniform, and it's not always the case. And also they use kind of a fancy media, which is, we wonder, does it really need that? In the end, you don't. Actually, this bacteria itself grows better on plate count agar than anything else. So, why they didn't start off with the easiest, I have no idea. All right, and then kind of one of our goals moving forward is to try to find a pretty easy way to do is take some vegetables that are starting to rot from my refrigerator or wherever, put them on a windowsill, take a wire loop, streak some from each of those onto a carrot slice, and then if in fact those are soft rot bacteria, you'll start to see the soft rot caused as those bacteria with their pectolytic enzymes melt away the pectic polysaccharides that hold those cell walls, those cells together, melts those away, you get the soft rot. So then all we do is kind of like a baby version of Koch's postulates. Take those, purify them, check them again, and then freeze them down. So we've got some good hits on the bacterial side so far. So just getting the bacteria is not really what we want, and ultimately what we want is then to isolate phage. So take from compost bins, soils. We have a soil farm on campus, south of campus, which is really nice. So take samples from that, use the filters, get out of here. Hopefully we have phage in there, and then see if we can find good plaques to show that we have now phage that are killing these soft rot pathogens. And that's the ultimate goal. So that hopefully in the end, we have cocktails of these phage that we can then use in a setting to minimize the damage for post-harvest rots. So about 15 to 30% of all crop loss is due to post-harvest rots. Much of that's bacteria. In tropical parts of the world, it's up to 90%. So it's very cheap, very straightforward method. Might be a benefit in as far as production and retention of those places that don't have really good cold, dry sort of storage. All right, so how am I tying this aspect of phage discovery into kind of a more long-term way to look at stuff with students? So as you might imagine, a lot of students, as they start off in the lab, have lots of common sort of problems. Like they streak out a plate for bacteria, and you end up seeing this the next day. Clearly there's something wrong. They either didn't sterilize the loop, or something didn't work right. Maybe they contaminated the solution. Or just like pipetting. Right? You got this nice pipetter, you want to move a mill to something else. It's not always obvious to a student that before you put that in a liquid, you need to put a pipette tip. So it's a benefit to do that. Other things like autoclaving, so I try to do a pretty good job because autoclaves can be a little dangerous. But if students in a pinch, they've got to go to class soon, so they decide that, oh, my media will be just fine. I'll just put it on fast exhaust. So, and then the media boils out, everywhere into the autoclaver, into the autoclave bin, that sort of stuff. One of the toughest ones for students is for serial dilutions, not just the math itself. Sometimes the students seem like they get the math and they understand the process, and then they're, very undiluted, 10 to the minus 1 or whatever, so no bacteria at all. But then after they're 10 to the minus 8, super diluted, they have a complete lawn of bacteria. So like, what's taking place there? So these are all things that I'm sure I've done most of those myself, but all those things are trying to think of, we come up with all these different possible things that can go wrong, and then come up with a way to develop some tools for students to be able to more efficiently learn these things with minimal mistakes. So I'll just zip through some of the others as I was setting this up. How do you use a laminar flow hood? Do you know how to use microscopes, Bunsen burners, water baths? Water baths work a lot better if they have water in them. Spectrophotometry centrifuge. Paraffin, you actually have to peel this little paper off it. And serologicals. I think what really confuses students is serologicals have numbers going in both directions. So if you want to pull up 4 mils of something and you see a 4 down here at the very bottom, you're only pulling up 1 mil, so you have to turn it around to see the rest of the numbers. So the hope is we can come up with some tools to minimize these sorts of problems for students starting off in a research lab. All right, I digress on that. So I'll finish up with some of I think some of my perceived biggest benefits of the position. So a lot of teaching, so I enjoy teaching, so for me that's a benefit. Obviously not everyone agrees with that, but I find it to be pretty fun. Variable research projects, right? Working on things from phage to grain leaf farms to research ethics stuff, so you have a lot of variation in the research, which can be really fun. Keep things sort of fresh. Also, along with that, lots of collaboration. One for here. So, of course, I'm expected for grantsmanship to write for grants, but I'm not expected to be a professional fundraiser. So as I was a postdoc, my boss, who's now tenured, he literally would spend 95% of his time writing for grants or editing manuscripts. Which for him, he had to, at a medical school, he had to raise over 90% of his salary forever, for the rest of his career. So that's all he does. And he literally couldn't find anything in the lab if you were to ask him. Also, I like the flexibility in the summer. You can do different sorts of projects. And probably the most importantly is the vast amount of support that people on Plant Path and Micro have given me to get everything set up and try to get the program up and running has been a huge, huge benefit. All right. A couple of the areas where I think there might need some extra attention or caution is probably just of the awareness of this type of position, 80% teaching, 20% research in the total field. I don't know, even 10 or 15 years ago, how common that was, if at all. So even, just to raise awareness that that sort of position might be of importance. As well as documenting your teaching. So if I'm 80% teaching, and the only thing I rely on are my teaching evaluations for courses which are service courses for people who don't want to be in the course, it might not be the best idea. So all the different programs with SELF for the things like the Teaching Partners Program and all of those other things really help to build up your portfolio of your teaching sort of skills and ability. So very important there. One thing that kind of relates to that is for curriculum improvement. I think this is kind of a cautionary thing, is that it's well known that if you make a major change to curriculum, and let's say I take my normal traditional lecture format class, and the next semester I make it a flipped classroom completely, your evaluations inherently are going to be much lower. Hopefully they'll raise back up over time, but it's been shown through lots of evidence that if you're going to make curriculum improvements, you have to be aware that you're going to have those sorts of problems along the way. So you have to balance wanting to make big changes to courses to try to improve it, and also recognizing that if you try to do too much to too many courses at the same time, it's going to lead to all sorts of problems as far as ultimately your documented excellence. And also research with relatively limited funds. I think there you just have to be, for me, more creative and try to pick projects that don't take a whole bunch of money is really the real key more than anything else. And hopefully we'll get some more funds for all that fun stuff shortly. And I'm running over a little bit. So I'll finish up right now. I'm in the middle of the third-year review, which strikes me as basically a time point to kind of check in with the department and see where am I at, how have things gone so far, what do I need to improve on, ultimately putting things together for the tenure package, just so we can make sure that all the ducks are in a row so that nobody's surprised at the time when it comes up for the tenure review itself. And then I was going to tell you at the end one of my big plans for hoping to renovate the Plant Pathology course, but I'll save that for another talk. So I'll take any questions if anybody has them. Any questions on that rambling dialogue?
Yeah. When did it occur to you, you have a pretty, impressive research resume, when did it occur to you that you would prefer a research career, or when or why, over a primarily research-dominated career?
Yeah, so that's a good question. So why ultimately did I choose to have principally a teaching sort of a pathway? Why did I choose that? So I was obligated to teach as a master's student and as a PhD student every semester just for funding. And I actually started to enjoy it. And then as a postdoc, I was kind of an interesting story. As a postdoc, I was the only person in our lab who wanted to seek out teaching. So I sought out teaching a medical micro lab for second-year MD PhD students between Harvard and MIT. It was really fun. I really enjoyed that teaching. At that time, I was the only person in our lab who wanted to seek any sort of teaching out. But at the time, I'll say this, and then at the time when I actually applied for this job here, there were more people on my floor, just on my floor, that were looking for research or R01 microbiology positions than there were microbiology positions in the entire country open. So it's a really limited field, and you can only imagine if there's that many, clearly, right, there's 1,000 times more qualified people throughout everywhere in the world. And it's to really kind of balance that with, yeah, there's that much difficulty and that much limited funding, and I enjoyed the teaching. I did not want to be ultimately just a career writer sitting behind my computer. And so that's kind of an iterative process, I think, over time. But I don't know. I feel like I'd get bored if I was just writing all the time. Yeah, go ahead.
But yeah, you have to write some. So I know you have only 3 years to look back on, but when you look at the funding scene for SOTL type research, do you see it increasing?
I don't know that I see any funding of anywhere increasing. Like I say, for the SOTL field, you have avenues in which you can find a niche that you might be interested in, and which you can also collaborate with people. And like we have a collaboration right now with Roger, we'll see if this Barley grant goes through. It would provide funding for multiple grad students for several years to develop a whole bunch of interesting central dogma sort of resources. So you can kind of find ways to collaborate with people doing broader impacts. As far as just raw SOTL sort of money goes, I don't think that there's any. Nancy, do you know of any increase, really?
I don't know if there's an increase, but there isn't a decrease. And I found, working with, like the IU's Innovations in Undergraduate STEM Education, that branch or that division of NSF has been pretty strongly funded traditionally for the last 8 to 10 years since I've been writing grad school. And yeah, the idea of collaborating for broader impacts and stuff like that, it works pretty well to be able to put in information in education and assessment and all of that for somebody else's research grant as well. So, yeah.
And hopefully it's reviewed favorably that you're trying to find people that like to do that sort of stuff. So.
Well, IU's has about a 20% funding rate right now.
That's much better than, I don't know if anyone else applying for funding higher than that?
5%, 4%, 3%?
Yeah, that's what
But
it was. Well,
the grant review
$100.
panel, on panels, it tends to really help. I mean, when you have not just I'm going to have people in my lab to do things, but actually have focused attention on improving teaching, I think it helps.
Right.
Yeah.
Just to follow up, casting your mind back to when you were younger and thinking about it, what would you recommend for students who are contemplating competing for a job where teaching is a substantial component, not necessarily 80-20, but whatever, where that's a substantial part of it, to make themselves competitive? What would you recommend?
Yeah, I think I had the same advice as, I forget her name, she knows the cranberry stuff in Massachusetts. Yeah. And she actually said at a seminar once is that the key thing to do is any opportunity you can to broaden your horizons, to teach or do extension or whatever you're interested in, take that opportunity every time. Like I was in some respects blessed that my postdoc advisor, he wasn't getting the research time out of me when I was teaching his course, but he thought maybe that can be of value. And so right now his lab has 13 postdocs, 12 of them teach at least a little bit in these liberal arts colleges around Boston. So it's changed. People are taking more of a focus on that as I'm sure general statewide and other funding changes over time. The importance of teaching seems to be on the rise, I'd say. So I'd say take every opportunity possible to broaden your horizons. It can never hurt. You never know when some random person you're talking to or interviewing with, and they're like, oh, I used to do something like that too, or I worked with that person once. I think it always helps.
Yeah. Other questions? No, thank you.